for coming. This is being recorded. For those of you online, if you have questions, feel free to ask in the chat and we'll come to those at the end of the talk. But today we're very lucky to have Susan Brawley, who's a professor at the University of Maine and has been doing research here for a long time. And so we're one of maybe the longest current ongoing research in the park, I think. And so look forward to hearing what you have to present today. And and thank you very much for making the time.
My pleasure. Thanks for inviting me. Okay, so have all of you used the University of Maine's Climate Change Center Climate Reanalyzer? Yeah, Sean Burkle's tool. Have you used it before? I'd be happy to show somebody afterwards, but it's really wonderful and it allows you to get daily sea surface temperatures in real time and to add them iteratively. So here, for example, it'll just take me a minute to adjust it. Okay, so I captured last week the mean and the standard deviation of the mean, which is the dashed lines, for the 1982 to 11 sea surface temperature in the Gulf of Maine. And of course, that's just the skin, right? Because the remote sensing doesn't go deep. So this is sea surface temperature for sure. And I picked the year 1991. You can go in and from 1981 until right now, today, you can bring up sea surface temperatures. And you can do it in centigrade or Fahrenheit. You'll see centigrade on my stuff. And you can change the box here to reflect smaller regions. So it's very handy. And Sean Burkle, our state climatologist, is in the Climate Change Institute, is really great. Okay, anybody have any idea why I picked 1991 as something to put up? It's because it's when I came to the University of Maine. And so from 1991 until present, I have spent a great deal of time doing research in Acadia National Park, especially at Scudic, but in other areas of the park too. And you can see that 1991 was a pretty good year, it fell right on the mean. Now, as we know, things have been changing, and Sean and I developed this concept of bioregions along the Maine coast in order to be able to look at that better. And they're based on my understanding of the algae. In other words, the dominant algae on the shore, and they're different here from here to here. And we marked the bioregions based on that. So it is somewhat subjective and also subject to change. And in fact, we're going to revise them with another paper we were working on, and they'll edge up a little bit. But you can see that here we are in bioregion 2, right on the edge of bioregion 3. So this dates from 2019 at the end. Again, this is centigrade temperature against years. And this is the sea surface temperature in bioregion 3, bioregion 2 in green, and bioregion 1 in orange. And so it's quite clear that the southern coast of Maine is warmer, and that they have all been trending warmer except possibly bioregion 3, which is pretty stable. And things have been getting worse in the last few years. So I left the 1991 on very blue. 2012 was our first really bad marine heat wave in Maine, so that's an orange. And 2016, which was also a heat wave, is in green. And 2023, which was pretty warm too, as you can see, is in gray. And then this year through about last Friday when I captured this. And so things were right down on the middle nicely for this year, despite our El Nino, and then it took a lurch. So I haven't looked at it the last few days, but you know, we're likely headed for another really bad summer. So that's obviously one of the things that is a concern to us, and I should go back to the second. Notice that falls have gotten much warmer. And August is the warmest month. Traditionally, March is the coolest sea surface temperature, but that might not continue.
Is 2012 the year that had the like early shedding for the lobster?
I don't know. But it was a really bad year. It was our first real repeat like in May. So here's what I'm going to talk about. I just want to review quickly the factors that affect intertidal algae and for that matter, most other parts of the intertidal community. Tidal level, desiccation, you know, not drying at low tide or freezing in the winter at low tide. Temperature, both air and seawater, nutrient availability, quality and quantity in terms of light, water motion. And those are all physical factors, obviously, but then herbivores, predators of herbivores, and positive or negative microbial associations are also important things affecting the intertidal algae. And so my focus for today is on two groups of organisms that I've worked a lot on. The fucoid algae. We have a number of species of fucus on our shores that I'll talk about, and a group of Bangiophyte red algae that are in the genus Porphyra. Most of them talk about Porphyra umbilicalis. And what I want to do is to tell you enough about the biology so that I hope you will be thinking about these factors. And as I talk about the biology, if one of these factors changes, what is that going to do to the biology and place on the shore of these organisms? And then, when we get to a point at which you want to start discussing things, no matter where I am, let me know. And I will quickly end things if I don't too long. So one of the things we can talk about is organism shifting zones in the intertidal zone, which has become very visible to me compared to what I'm used to over the last 3 or 4 years. And again, I want you to be thinking about how some of these things, as they change, will change the shores of A&D. And here is Porphyra, the red alga, and then there are just massive amounts of fucoids, and at this zone, it would be the mid-zone fucus specificulitis. I'm not going to have time to talk about the microbial study, but about 16 people across the North Atlantic that I organized between me and my equal PI at the Marine Biological Lab, Lily Morrison, have been studying the microbiome on Porphyra umbilicalis and on fucus speciculitis. And these are our study sites. And we're just going to obviously focus on Scudic today, but at some point in the future, I'd be happy to come back and talk to you about that. I did something weird. Seems like I rotated something. I'm not gonna worry about it. But anyway, look at this beautiful view of Scudic, which one of the 3 papers we have published out of the microbiome project got put onto the cover of Journal of Phycology. And here, what I want to do is just to show you the 3 main fucus species. And so Fucus spiralis is the smallest, and it is in the upper part of the intertidal zone. And these sort of bulbous ends are called receptacles. It's where the gametes form. This is fucus vesiculosis in the middle zone, and it has a very broad zone on our shores. It has air bladders, except where it's really rough, and then it doesn't develop air bladders. This is a male. Both sperm and eggs are produced in these two species in their receptacles. So they're separate male and female individuals for fucus. And I know this is a male because the receptacles are orange. Now, if I cut one of those conceptacles, this is what I see. And this is from the fucus vesiculosis individual that was a female. And so there's all this gooey stuff in the middle, which is actually an important part of the force that with ionic changes pushes out the gamma-tangent. And, you know, many organisms, including the fucus species, have external fertilization. So the wild and woolly sea, how successful is it? There have been many studies published in which people have said that sperm are limiting and external fertilization is very inefficient. And our work shows that that's not true, and some Syrian people, a different group, have also shown that it's not true. And it makes sense because if you think about what the force of selection is, if something can't get fertilized, there's no further evolution. And so anything that allows fertilization to occur under unfavorable conditions should be favored. And so not releasing your gametes under environmental conditions in which they're all washed away would be a prime example. And we're going to see something about that. This is a packet of 8 eggs that comes out of the conceptacle. And very quickly, there's a little polysaccharide envelope that dissolves and the eggs are released. But in the lab, if you add sperm, which I had done, and that's the eye spot, they will quickly be attracted and they manage to eke their way in, and eggs are probably fertilized before they actually are in the water. This shows an electron micrograph of the sperm with one of the flagella of the sperm resting on the surface of the egg. And this is the conjugated octotriene that Meyer and Mueller described as the sexual pheromone that attracts sperm to eggs. So the eggs release this nice perfume that attracts sperm. And it's very effective, as you can see on the next slide. Okay, so here fertilization has occurred. The sperm and egg have fused. Here's the sperm nucleus as well as its mitochondria, and a bit of flagellum up there that has been taken into the egg. And now that has to get from there to here. So there's the sperm nucleus, there's the egg nucleus, that's the nucleus of the nucleus. So the sperm is probably here, and it had this nucleus had to migrate in to fuse and form a zygote. Now there are lots of interesting things about this. First of all, when we collect materials in the sea and we fix and stain them, we can absolutely tell whether fertilization has occurred because we're seeing the sperm nucleus. We can also tell whether only one sperm fertilized or more than one sperm, which is important, we shall see. And then I realized a bit down the way when we were trying to do experiments to know when during the tidal cycle fertilization actually occurred, that this migration, which takes around 2 hours, could be used as an internal clock for when fertilization actually occurred. Now you have to set it up in the lab, right? Same temperature as the seawater and all that stuff that you can imagine needs to be done. but you can calibrate fertilization to any natural situation. And so you'll see that in some of my studies. Okay, this poor Fucus vesiculosus egg was fertilized by one sperm and another sperm. This is the egg nucleus again. So this sperm nucleoplasm has spread already in the egg nucleus, and that one's probably just freezing, but it's gonna die. Now, of course, I killed it, so it already died. And the reason that is called polysperm, the reason is that the extra genetic material is not a problem, but the centrioles, which set up the divisions, like the first mitotic division, they are inherited through the sperm. And so if you have two sperm fertilize an egg, you've got two sets of centrioles. And the first mitosis, there are weird, unusual spindles. And so the chromosomes get pulled every which way. And pretty soon, first one cell, and then as a couple more divisions occur, all cells are missing some housekeeping genes they have to survive. And so it will kill the zygote. And it's true for most organisms. Birds, lizards, they do something a little differently, and I can tell you about it later if you want. This should not be like that. I seem to have. Do you think if I push that here, will it recenter?
No,
Probably not.
I think it's probably.
I think I'm just gonna go on, but anyway, this graph, which is actually sort of important, has two different ratios of sperm to eggs in the laboratory experiment. And it has from a normal concentration of sodium in seawater, this is an artificial seawater, which is 450 millimolar. It shows whether in fertilizations it's good, there's only one fertilization, or it's very bad. And since you can't see this, I'll just tell you that as you increase the sperm egg ratio, not surprisingly, polyspermy goes up. And it gets very severe when the sodium has fallen to 45 millimolar. And these experiments are done with something else added as an osmoticum that is physiologically neutral, but can replace the osmotic effect of sodium, which is obviously huge since we're talking about seawater. You can't screw that up. Wasn't that? Well, okay, so at 2 weeks of age, after having germinated about 15 hours after fertilization, here's the rhizoid, which then grows out. You have something called hair cell stage. The hairs will fall off, but a lot of polysaccharides being secreted in the rhizome, because of course otherwise these things are going to be washed away by waves. Although a lot of the zygotes settle down into like little crevices or barnacle crust, so that's an important recruitment issue for a few of the species. But here's some polyspermic embryos, same age as these, and you can tell that they're in very bad shape. Most of the cells are dead, and all of them will be. Okay, now, how does an egg prevent more than one sperm from fertilizing it? It has to be fast under a lot of circumstances, because contrary to what is the default, sperm are not limited in any of the studies we've done, even in the Baltic. And so I'd just like you to look at these traces, which are recordings of the membrane potential inside an egg. And the ones that are under A are different places, different species, although Ficus vesiculosus is A through D. And these are ones that have experimental lowering of sodium, or in one case, Ficus, let's see. But they're all experimental. So, the main point is sodium is limiting in these as opposed to this. This is like being fertilized in normal seawater. And so if you look at this little spike, there would have been a little spike a little bit ahead of where I cut off those traces. But that's when I dropped some sperm into the bath, where I had a microelectrode inserted into the egg to record its membrane potential. And there is, as soon as the sperm fuses with the egg membrane, there is a rapid depolarization. This, you can see the scale of the millivolts here. Typically, the resting potential of many cells, including Fucus eggs, is around -60, -70 millivolts. And so there's a very sharp depolarization, which is caused by the entry of sodium ions. And that lasts for about 5 or 6 minutes under normal marine conditions. But when you have lowered the sodium, so sodium's limiting, and think of an estuary or something like that, you get a very slow rise in the fertilization potential, which is dangerous because there's sperm out there. And all they need is for the fertilization potential not to achieve its rapid rise and hold. We think that this actually has not been shown in any system, but the model is that the sperm and egg receptors are charged proteins, and they have to fit like a lock and a key. If you change the membrane potential, you can change the shape of molecules. And so you can have literally hundreds to thousands of sperm sitting on a Fucus egg, and none of them can get in during this fertilization potential except for the one that triggered it, that opened the sodium channels so that sodium could come in. So that's called the fast block. And then cells aren't meant to be at that kind of membrane potential and screw them up. So another mechanism evolved to be a permanent polyspermy block, and that is cell wall formation. So I think all of you know that brown algae have a lot of alginate, this polysaccharide, and you put it in ice cream, paints, and other things, right? So this is a stain called capriflor white that stains eggs when they are fertilized. And so I had dropped some capriflor in, and these others would soon have been fluorescing. But it takes around 3 minutes for the slow block to become effective against polyspermy. So you have the two working together. And I know some people are interested in ascophyllum, so I just want to show you some ascophyllum potentials. And this is marine eggs, south of Bucksport, where it's really pretty saline. And this is 2 kilometers up from Bucksport. And I collected Penobscot River water and ascophyllum to do both of these experiments in the lab. And so you can see that fertilization potential in estuary water still rises well, but it probably doesn't last long enough to protect the eggs. And secretion is also slower under lower salinity conditions. So you would anticipate that there would be higher polyspermy there. Okay, so we had done all these experiments. Every place that I had been, I had found a fertilization success by that staining technique and collecting the gametes in nature, the eggs. That fertilization success was between 98 and 100%, and very close to 100% in terms of the actual mean. And so we wanted to do some experiments, which we did at Pimlico first, and then subsequently we did at Scutum. in which we were capturing sperm and eggs from a water column to see what the ratios were in nature. And I made monoclonal antibodies against fecus sperm so that we could detect them. Because fecus sperm look a lot like a lot of phytoplankton. There'd be no way to, under a microscope, say, okay, this is a fecus sperm. I'm sure that I'd never walk a plank on that. And so this has a fluorescent dye attached to the monoclonal. And here's the trailing flagellum of the fecus sperm in this area, which has lots of tiny hairs on the interior directed flagellum. Really lights up. And that is the part that first attaches to the egg membrane. So it's probably, probably detecting with this monoclonal a lot of receptors. And so that was good. So this is the Dariguagas estuary. Does everybody know where Dariguagas River is? Have you been there? Mm-hmm. So it's fairly shallow and the tide comes in quite nicely. And if you're driving on the North Road by the river from, the entrance at the bridge, you have to go up about 4 miles maybe, and then there is a rocky shelf that's quite large that is covered with Fecus fasciculosis. Now, I haven't been there for probably 8 years. It was for a long time. And so it was a perfect place to do these experiments because it was all Fecus fasciculosis, nothing else interfering. And so we had devised these pumping stations to use to filter the water and then be able to fix the samples in the field. And so the pump was attached, each one was attached to 3 hoses. And so it went out for maybe 20 meters or so and was attached to the bottom in different parts of the bed so that we get a good sample size. And what we found by wiring things up too is that the photon flux density, micromoles meters squared per second, was of course high at the beginning and end of the incoming tide because the bed was exposed. And then our hypothesis was that the gametes would be released near the highest point of high tide. Anyone have any idea why that was our hypothesis? High salinity? Yes, that's a very good point, because we certainly would have high salinity. But in this particular area, the shelf is really covered by saline water and not in low salinity water. We have to go farther up the river to have that issue. You've heard the phrase slack high tide. Mm-hmm. So we figured that at slack high tide, if the gametes had just been released before that time, fertilization might be very high. And so these are time intervals in 30 minutes, and we were collecting water samples in Dariguagas using the same apparatus at these blocks and staining any eggs that were there. But we didn't find any eggs until or sperm. You see the sperm come up just maybe half an hour before the eggs. Nature's very clever. And then the peak was, let's see, the peak was perhaps 30 minutes to an hour, that interval after high tide. And so we have a sperm egg ratio, and we checked the sperm again with the monoclonals. We had a sperm egg ratio that was about 10-to-1. And that was quite adequate for all the eggs to be fertilized, 99 to 100%. And in our filtration, the fixation is done immediately, so nothing's sitting around. And also, of course, we receive the pronucleus inside the egg later in terms of our timing. So this was really very cool. So that told us that calm water was important and that light was important. This is Jessie Newland, and this is wonderful work she did, and some of you will already know about it, and you should ask her to come talk about this work if she hasn't before. And I'm not going to take the time for that, but I'm going to show her next experiment, which was published in the Ecology Progress series, because this is a scuba. And here you can see that we were sampling at 4 different locations around Scoot Point. I have to say the one-way traffic circulation was a real pain. And the time that I locked myself out of the car when it was about 5 degrees, when I was at the site just north of Blueberry Hill. That was also aggravating. But we got some great data, and again, time is limiting. So I just want to make the point that light, the photosynthesis, and calm water require the sperm and egg release to occur. So just focus on, I'm going to ask you to focus on a few different places. So first of all, you can see that whether it was sunny or cloudy is represented just subjectively. I mean, we wasn't subjective, we had a radiance reference, but for this graph, it's subjective, right? So full sun is 3 and really dark is 0, even though it's daylight, cloudy or foggy. And so you can see that here, it was really not a very well lit day around Scudic. And the wind was also strong and coming from the south. We didn't capture any gamete release. There are other times, like, both of these, you can see that here there are 3s around the point. And so at each of our 4 sites, we saw a lot of gamete release. And this one is interesting because the wind was coming from the north, I guess the north, right? Close to the north. And you can see that it was very sunny in one site, which is where we got a lot of gamete release. And it was basically cloudy in the others, and there was no gamete release. Actually, that's not quite true. Here it was sunny, so I can't really explain that. Yeah, and you can see that these graphs also show the strength of the wind, so there are barely any lines, regardless of the direction. It was calm. So from these data, we could infer that a sunny day and calm conditions were recorded. And of course, in this case, it would have been very rough as well as cloudy. So basically, natural selection has hijacked photosynthetic metabolism to trigger gamete release and calm seawater. And that is true, even though you can have 3 weeks in a row without a calm day. And then the next calm lit day, we'll get a massive release of gametes. So how to avoid algae since water emission? We did this with a different alga from the Pacific coast that I worked with because it's a very good lab system. And basically, if you put the receptacles in seawater in the lab for 4 hours or more and then turn off the lights, gametes rain out. So again, there's a photosynthetic potentiation of gamete release. DCMU is an herbicide and it inhibits photosynthesis. And so if you look at this experiment, this is a control. And so there wasn't any inhibitor and the receptacles were potentiated. And when we turned the lights off, all this gamma release occurred. However, when we added DCMU to these replicate beakers with receptacles, and then turned the lights off, there was no gamma release. And if we didn't turn the lights off, but we just added to this replicate batch of beakers DCMU after 7 hours in the light, they released gametes. So it was shutting off photosynthesis, that was the trick. You could do it with the lights, you could do it by putting in a photosynthetic inhibitor. And this is really the explanation of why calm and turbulent conditions make such a difference. And so just for a moment, imagine yourself as a fucoid alga hanging out on the shore. And it is a calm day. You have to take up CO2 to carry out photosynthesis, to grow and all that stuff. And in the brown algae, there isn't a direct uptake of CO2 because it's very limiting at ocean pHs. There's an enzyme called carbonic anhydrase that sits just on the outside of the plasma membrane. And it takes a bicarbonate ion and it extracts CO2 from it, leaving a hydroxyl ion out there. And that immediately passes over the membrane because carbon dioxide is small and not charged. And so you're happy, right? You're chugging along. And then suddenly, because it's calm, the boundary layer around you gets thick, which is to say that you've taken off all the bicarbonate that was near you, and you can't get any more. But if the conditions are turbulent, of course, there's still a boundary layer. It's always a boundary layer, but it'll be a minuscule width around the fucus individual compared to calm conditions. And so Derek Pearson, the Esters of Howe, who were in my lab, very cleverly did this experiment. So in each of these conditions, we're putting receptacles into a beaker, and we have them in the light. And some of them are under just calm conditions, and others are being agitated on a shaker, simulate water motion. So in the calm condition, this 2 millimolar bicarbonate that we added to the seawater simulates the natural condition. And so at the end of the period of time when you knew that it was being potentiated by the light, you turn the lights off and the love gametes release. In that same treatment, the agitated receptacles did not release many gametes. So we found that by manipulating the bicarbonate concentration, either to start them off immediately as if they were under long, calm conditions where they were starved for bicarbonate, or continually agitated, we could manipulate the gamete release. So here under calm conditions, the gametes get released in large amounts, but under agitated conditions, you see they also do, because the bicarbonate sensor isn't there. It's already zero bicarbonate, and the stirred medium isn't relieving that. It's not bringing more bicarbonate to the surface of the receptacle. And under calm conditions, if we took the agitated treatment and put those replicates at such a high level of bicarbonate that they couldn't possibly deplete it, then when we turned the lights out, there's still no release of material. So just manipulating the bicarbonate concentration to reflect weather conditions in the tidal zone, calm or being stirred or, waves going like this. That did the job.
Do you think 5 or 10 minutes? Would you be able to wrap up in 5 or 10 minutes?
Yes, I can. And what I'm going to do now is move to talk about porphyra. And so there's a lot of porphyra on the shore. And this is a cutaway of different sections of the blade. You can see again, they're rhizoid cells and they make a lot of good. Nick Bullen in my lab discovered that in the northwestern Atlantic, as far as we can tell, and we've looked at probably 20,000 blades at this point in different areas, there is no sexual reproduction. It's only reproduction for neutral spores. But in the northeastern Atlantic, there's a sexual life history. And some blades also reproduce asexually. Now, this could be a little tricky. This is a film. Okay, I think I can trigger it here. Okay, so the shadow is a piece of a blade in a dish. And those things on the bottom are fluorescent red because it's the chloroplast, and the chlorophyll, when it is illuminated with blue light, will fluoresce red. So that's the setup. And none of the red algae have flagella in any form whatsoever. People have thought for a long time that they were just helpless, I mean, they're a really robust group of organisms, but they didn't seem to have any way to figure out a better recruitment spot than another, even on micro scale, because no flagella. And so, look at these neutral spores. Do you want to see it again? So these are individual tracks over 120 minutes, compressed to 1 minute of runtime, right?
Yeah.
And now I'm going to show it to you at a higher magnification. This is Josh Kelly, one of my colleagues on this project is at Maine. And the wonderful thing about this is that it really shows that the neutral spores can move to the right level of sun and everything else. They're really lovely, aren't they? Now you see that one is really moving directionally, and these two are just jiggling around. And that jiggling around can be important too, because that kind of behavior can allow a neutral spore to get out of the gooey boundary layer of a blade. So, we've characterized that. And they germinate and they make all this goo that's, down in that area where you see rhizoid cells. This is the chloroplast, lots of vesicles that are being secreted. It's highly sulfated, which I know from that dye. And they do need to stick really fast and hard because the tide's coming in. Okay.
Washed away.
Yeah, and so here is porphyra, scudic, and Litorina. I've been taking the digestive tracts out of these and making crude enzyme prep to digest the cell walls of germlings so that we can get labels in to stay the cytoskeleton. And that's a story for another time too. But what I found was that these are brown and white. This had been in ethanol. But there is one particular amphipod that in the spring really starts to chow down on porphyra because it's beginning to be stressed and is not growing well. And so it's a sort of city prey, but still very nutritional for the amphipods. And so this one particular amphipod does that. And you can see that instead of having lots of little neutral spores ready to release, it's a raised edge. caught up with me. And that's not been pointing at.
What battery would be down, sorry.
That's fine. I mean, I've got to stop pretty fast anyway. Let me just get a highlighter too. So do you all know that brown algae are not plants? I don't know why this isn't working. Oh, there, okay. So this is what we think of as a tree of life now, although it's not rooted. And here we and the fungi are, that's called the hispicons. And here are the diatoms and brown algae and a lot of other things like that that are in the sternum piles. Here are the plants, although in our original figure, this is a dashed line because in fact, it's not absolutely certain this is a monophyletic group. But here are the red algae, and the green algae, and that little group called the land plants that all down them. So it's okay to call the red algae and the green algae plants. I mean, they're part of that general group, but not for brown algae. And maybe I'll have to end with this one, but the Porphyra umbilicalis was done, the genome, the nuclear genome of Porphyra umbilicalis was done from blades that I cultivated and cloned and grew in the lab to have the immense amount of material DNA that the Joint Genome Institute needed when our project began to do a nuclear genome. And it was a really big deal. It took 10 years to do it. And so we had some big celebrations. There were 50 scientists that I led in that project, some PIs, co-PIs. And that paper has all kinds of interesting biological stories because all of these people were specialists in different things. And we just learned so much. And I'm not going to have time to really talk about my part, which is on cytoskeleton, but it's ongoing, so you could have me back sometime in a word. Anyway, the reason it took so long was that the genome has a very high GC content. 73% of a protein coding gene, 73% GC in the protein coding genes. That's like trying to sequence kryptonite. It was simply impossible. And everybody was so frustrated because we spent so much time getting the samples to JGI. And then PacBio was invented. And PacBio sequencing is not sensitive to GC. And within about a year, we had recovered and got things published. And so those are my two co-PIs, John Sollenbacher, Grossman, Nick Lewin, my lab, Simon Broughton, who's an incredible guy. I was flash through and say you can get, oh, this is really important. Can I make one more point? I mean, this is really important. I probably could have delivered a link at the end so I could do this. So, red alkyl chloroplasts, in addition to chlorophyll A, have phycobilin pigments, and they are as much protein as the fluor that actually captures the light. And they are organized on the membranes inside the chloroplast. This is just one membrane, and there are lots and lots and lots in the chloroplast. You see it in the screen right now. And so erythrin is on the outside, and that is where it absorbs. Phycocyanin is next, and that's where it absorbs. And then allophycocyanin is in this area. And so this is the chlorophyll and carotenoid spectrum as a baseline, and then this is there. So what are red algae able to do with all these pigments?
Catch wavelengths that other things can't.
Right, and on almost everything, right? But they're moderated in amount, or the red algae would look black, but some red algae do look black. They're so dark and loaded with these. Okay, so the clincher that's a problem is that because they're protein and red algae, could you just stay 1 second?
Yes, ma'am.
That's Congress Christmas on the coast of France in 2004. It was the first time I had ever seen Congress Christmas turn pea green. There it is. So, when a red alga is so stressed by high light and especially by high temperatures, it has to break down the phycobiliosomes to recycle the nitrogen in that protein to make heat shock proteins. And the heat shock proteins wrap around all the other proteins that are being made and help protect them so that alga can live. Now, if this lasts too long, it's not going to be good. And it's also happening because this is in summer and the water has been stratified because of the heating of the surface water. So all the deep nutrient-rich water is not mixing up to the surface. And so nitrogen is limiting, stress is very high, and the organism would die except for this ability to recycle the phycobilisomes. And I have seen a number of red algae, including on our shores in the last few summers, including in a couple places, Congress Christmas turned green. So pay attention to it. It's just an immediate biomarker that things are really hard in energizing. I think that's last slide.
Thank you, Susan. I'm sorry I have to go to October 1 o'clock.
Yeah, you know what I said. So these are just people who have done the most recent work at school with me, as well as some who did work longer ago, a Canadian. And this is a frozen pool of a little dwarf fucus called Fucus disticus, the species disticus, that is found in the high intertidal zone. And remarkably, it reproduces in winter when it often is frozen underneath the ice. But what benefit does that have? If you're a fucus and you're reproducing under the ice, you know, at low tide, calm water. And also it gives things long enough to be fertilized before the water comes in with the tide and melts the tide pool and disperses things. So you'll see, you know, out on the shore, look for the small fucus, and that's what this is.
Okay.
But I have been, this is my colleague at MBL, and we're still doing massive amounts of work on our North Atlantic microbiome project. But it's very exciting, and we've got 4 more papers to publish and finishing the experiments for one of them. I think by the end of next week. And so it's been quite fun. I've just been so lucky to have so many good people.
Well, thank you very much.
Work in this wonderful place.
Yeah. Well, it's amazing.
Oh, from the park.
Yeah, it's an amazing amount of stuff that you, I hadn't appreciated how far back, like, it went to, just like the basic reproduction of, and fertilization, stuff that you were doing when you first got. That was awesome to see. Any questions? I know we're at the top of the hour, like 1 or 2 questions.
I was just gonna ask what changes you've seen since you've been working with it in the final.
The low zone until, I think 5 years ago, was still heavily covered by Fucus disticus subspecies edentatus. And of course, there was chondrus mixed in and mastocarpus above the chondrus, but it still looked like a fucus zone. And now it's chondrus and coralline. Fucus spiralis is a much smaller zone because our temperature conditions, as well as, you know, long light in summer, is really inhibiting Fucus spiralis. And so it is becoming rarer in its zone, although I can see hybrids between Fucus fasciculosus and Fucus spiralis because I know what those hybrids look like so well. I haven't sequenced them, but I know they're hybrids. And so the Fucus fasciculosus zone is still broad. And it's pretty much in the same place it's been, except the upper part is hybridizing with spiralis, and it will eventually, as sea level keeps rising, be an extension of the Fucus fasciculosus zone. Now, the Fucus spiralis doesn't have any place to go yet. If we were in Europe, and I brought this wonderful book so that anybody who had time could look up Helvetia canaliculata, because we don't have a Fucus like it in the Northwest Atlantic. But it's small, as you would expect, and it grows in ultra high intertidal zone in Europe. And it's so dry on neap tide series that you can just crush the whole thing in powder. But it can survive there. Well, we don't have anything like that here, either fucoid or really any other alga that can sustain that level of stress. Fucus spiralis is moving up a little bit, but into crevice areas and shaded areas of cliffs. And so we're definitely having zone changes. And I think in the actual transect surveys, some of these things are being found, right? I haven't heard anybody talk about spiralis, which I see, but certainly that Congress and Carolina moving up, I think is in those transect surveys, right?
If you're looking at me, yes, so Adam talks about the article that, there's an article in Quick Science, about Susan Dorff, so if you wanna read more about it, and Adam didn't talk about specific species, but he did talk about the subtitle, the shift in zone, and so a lot of the subtitle, inventory monitoring sites are like being flooded more. So there was more subtitle level.
You're talking about NBTN.
Pardon me?
You're talking about NBTN?
Yes.
Oh, okay.
And the kelp species are also really diminished. So the only kelp now in the intertidal zone that is at all common is Laminaria digitata. With some of these things, you have to be a little concerned about your ability to tell nature from the harvester who comes by and takes things that he or she shouldn't. But I think in A&P, there's very little harvesting by road people anymore. I'm not down here as much, but I could see it ebbing away. And I also talked to a bunch of people. They weren't doing it, but they thought they knew who did. And they talked to those people, and I think they stopped coming to A&P. Alaria, which I've worked on a lot, and is such a wonderful kelp and very tasty. It should be the main focus of kelp aquaculture in Maine. Not Saccharina latissima, because Alaria not only can supply alginate, but really good food. It's our equivalent of wakame, which is Undaria pinnatipata, which is the Japanese food kelp. So I can't find Alaria in places on the Scudic shore that I used to be able to find it quite easily. And Great Pool, I don't know whether you all know what Great Pool is, it's my nickname, but it's sort of well known. Great Pool used to be full of kelps, not anymore.
This is right off the Scudic Road where the stairs gonna go into the.
Yeah, close to it, yeah.
Out to Scudic Point, there's a little pull off of a little stair, there's a very big tide pool.
Yeah.
Yeah, no, any one last question? You good?
So I was wondering about whether you have been to Big Moose Island, Sunview Trail, and that intertidal zone, what your sense of that is. Because at the time we were doing the general management plan, Susan gained access to Big Moose Island, which was effectively closed by weed based it everywhere.
Yes,
And the Sunview Trail along that. Your comments about, at the time, you said it was the most pristine title zone in Maine, or the most that you'd seen. And it caused us to develop a policy in the general management plan to not promote the Sunview Trail, which has since fallen away from people's awareness, and I bring it back, but we at the time did not want a whole bunch of public use down there by the general public, that it was gonna be meant for student institute program. So I was just wondering if you,
I haven't been there recently, but I wouldn't imagine that anything bad has happened to it. I mean, the problem with the increase in seawater temperature is that Saccharina latissima is really sensitive to it. For that matter, so is Lavaria. But 2016 was a very bad year for Saccharina latissima in southern New England. Virtually all stands died. And, you know, it's been there thousands.
Do you think there's any value in, so after the storms, you can see a lot of kelp, you know, of just in the rack, there's a lot of kelp, and I wonder if there's is there any value, is that like a representative sample of what might be out sort of in the subtitle to like, is there any value in looking at the kelp that's washed up as rack in storms?
Yeah, I think especially in the citizen science-based way it is. But, you know, there's so many remote, ways to look at the shallow subtidal now. And between folks at our marine lab and, folks like Emmanuel Boss, who has a lot of gliders, getting him interested in doing glider passes in areas of interest out here would be worthwhile.
I think we're out of time, but
Yes.
thank you very much, we really.
I don't have a presentation.
No, I interviewed for the article.